Although Gregor Mendel's work greatly revolutionized the way that we understand the patterns of inheritance, there are some patterns that just don't follow the rules and don't follow the expectations that we would have based on Mendel's work. Now, we need to go back and remember that there were three assumptions of Mendelian genetics. First, that each individual carries two forms of the genes. We call those alleles. We assume that the traits are going to come in two forms, dominant and recessive, and that these two alleles are going to separate during gamete formation, and that that the alleles segregate. Now, some violations to this are some examples of human blood type has three alleles. So when we cross two heterozygotes, we're not going to get the 3-to-1 phenotype ratio of the offspring that we would expect. Another example of this is where we do not have a clear dominant or recessive pattern. So for example, these 4 o'clocks here, these little flowers, the red is the homozygous dominant, the white are homozygous recessive, but when we have our heterozygotes, they show up pink. And this is because they have what we call incomplete dominance, where the heterozygote individuals show a phenotype that's a intermediate or a blending between the two homozygotes. Another example is where we have heterozygote individuals that show both dominant and recessive phenotypes, like these asters here, that show both the red and the white petals. We're showing both homozygous traits. Another example is when our pesky little alleles of different traits are going to be living on the same chromosome. And so we call these linked traits. We're going to spend a little bit more time in the next chapter talking about sex-linked traits because those patterns of inheritance are very interesting. But an example of this could be hemophilia or color blindness, orange tabby cats. Those are all sex-linked traits where we see those traits show up more often in biologically male individuals than biologically female individuals. And we'll spend some time talking about why that is in the next chapter. Gene by environment interactions is what I actually studied when I was in graduate school. But this is where the phenotype of different organisms is going to depend on the genes that the organism has, but also on the environmental conditions. So an example of this, that's not a Cottonwood example, you're welcome, is the temperature, the body temperature determining the coloration of Siamese cats. So the pigment that produces the dark color on the cat's face, feet, and tail becomes inactive at over 33 degrees Celsius, which is the core of the cat has a higher body temperature. And so that part of the cat's body doesn't have that pigmentation. And so that's why Siamese cats look like that. Then when we're introducing this idea of alleles, we talk about hair color, skin color, or height, or things like this. But these are traits that are controlled by multiple genes. So this is called a quantitative trait, meaning that the effects of the genes depend on multiple genes, right? So it's not an either or; it's a continuation. It's a spectrum. So we can see a lot of variation in skin color, hair color, and height, and we know that that's typically because those traits are under the control of multiple genes. Okay, and then our last one is that this idea of epistasis, where different genes can interact. So we are actually going to have a dihybrid cross, but that's not going to give us our 3-to-1, our 9-to-3 to 3-to-1 ratio that we would expect. It's going to be more complicated than that because the effect of one gene affects how the other gene functions, and we can see that with the coloration of the coats of Labrador dogs, which they're just the cutest and the best little labs, the bestest dogs ever.